Hi, I'm Courtney Cheetah, and I'm the Program Associate with Wild Farm Alliance. Thanks for joining us today for the Whole Farm Sustainable Pest Management course. This is the sixth lesson of 12, and our mission is to promote healthy, vibrant agriculture that protects and restores wild nature. And we will help bring nature back to farms. Just a reminder that all participants are muted. If you're experiencing any technical issues, there are moderators in the chat that can help, and use the chat to ask questions to our presenters. Our 2050 vision is to see 1 million nest boxes and perches on farms, hedgerows of habitat the length of to the moon and back to help support all the different kinds of beneficials on the farm and restoring riparian areas because they are the lifeblood of the landscape. And we really see that avian pest control is a part of the toolbox for managing crops. We're excited to announce that participants can sign up now for our sustainable pest management listserv that will launch sometime before the end of the year. You'll be able to connect with others about sustainable pest management and learn about in-person online learning events, research, books, journal articles, publications, videos, and other pieces of knowledge relevant to sustainable pest management. And for those of you that are interested in learning more about sustainable pest management, Wild Farm Alliance is hosting a pre-conference workshop ahead of this year's Eco Farm Conference. We have a really great lineup of speakers for January 22nd, and the registration link will be in the chat momentarily if it's not there yet. Today's webinar is Managing Soilborne Pathogens Without Chemicals, and we have Margaret Lloyd from UCCE and Martine Guerrero from NCAT with us today. Next week, we'll be back on the 10th of December on Tuesday for our 7th lesson, which is coexisting with and supporting beneficial birds and bats with Austin Spence from UC Davis and Rachel Long, emeritus with UCCE. And please consider attending the rest of the lessons in the series. They occur about every 2 weeks through March. If you missed one of the earlier ones, they've been really great, and the recordings are all up on our website under the online courses under news and events on the wildfarmalliance.org website. We have continuing education credits available from DRP and ASA. And I'm going to pass this off to Margaret next. And if you have any questions, please put them in the chat. Or if you're having issues, let us know.
All right. Good morning, everybody. It's great to be here. Thank you for the invitation. My name's Margaret Lloyd, and I'm a farm advisor with UC Cooperative Extension, and I cover Yolo, Solano, and Sacramento counties. And this is one of my favorite subjects. I'm a plant pathologist by training, so it's always fun to focus on soil-borne plant pathogens. And today I'm going to just focus on 2 key areas of managing soil-borne pathogens, and that's keeping inoculum levels down and reducing the efficacy of the inoculum. So I'm talking about soil-borne fungal pathogens like southern blight, fusarium wilt, and the other fusariums, charcoal rot, and verticillium wilt, just to name a few. And when we talk about plant disease, we often reference the plant disease triangle because it's not just the presence of the pathogen. You have to have the pathogen to have a disease, to have that disease, but you also have to have a susceptible host and the right environmental conditions. And so to manage disease is to manage all 3 of those components: the pathogen, the plants, and the environment. And so that includes keeping inoculum levels low, that's the pathogen, maximizing plant health, and creating a less favorable environment for the pathogen and a more favorable environment for your plant. So to start our talk off today, I want to review a disease life cycle. And it's always important to know the life cycle of the disease that you're talking about or interested in managing so that you know what the intervention points are. So if we start down here at the bottom, we have the pathogen. In this case, I'm just doing Verticillium dahliae. It's a soil-borne fungal pathogen with a host range of over 400 plants. It affects many crops. And so it propagates and causes disease with mycosclerotia. And that's what you have here. It germinates, it grows towards the plant root, it tries very hard, it makes many, many attempts to penetrate the root tips. Eventually, one of those mycosclerotia will successfully invade the root cortex and grow into the xylem. And at that point, it becomes a systemic vascular pathogen that then travels through the xylem and causes wilt. That plant then becomes infected with the pathogen. And when the plant dies, it signals to the pathogen to start creating reproductive survival structures so that when the plant dies, it falls to the ground, all the inoculum gets distributed into the soil, and it's ready for the host to be planted again. So, as managers of diseases, our opportunities to interfere with this life cycle are at the beginning to interfere with the colonization of the plant and before the plant dies and becomes infected with the overwintering propagules. So that's why roguing is a technique, roguing out dead plants before the inoculum forms so that you prevent that inoculum formation and or removing infected tissue from the farm. Once you have your tissue, then it lands into the soil. So you want to try to prevent infected plant tissue from getting into the soil, from surviving in the soil, and then you want to prevent it from reaching new plant roots. So this is the zone that we're focused on when we talk about managing plant disease. And here are what some of those overwintering structures look like. So in verticillium, these little black dots are the mycosclerotia. That's a cross section of of like a plant stem. Here you can see these little perfectly spherical balls. That's southern blight, sclerotia. Here under a microscope, you can see Fusarium chlamydospores. So a grower calls you and has a disease issue. Say, hey, Margaret, I've got all these plants dying. Come out and help me figure out what's happening. So you go out there and you say, and you figure it out. Okay, you've got Verticillium dahliae or Southern blight or whatever it is. This is Southern blight in this case, this picture. And the grower says, well, I've never had this before. And then they always want to say it came from the nursery. But what I want to point out is that the first time you're seeing this is usually not the first time that that pathogen was introduced into your field. Because most soil-borne pathogens have inoculum thresholds, okay? So I have a schematic here of the inoculum threshold for Verticillium dahliae in lettuce, okay? And so this is 0 inoculum, and here's over 270 mycosclerotia per gram soil. That's how we measure the density of mycosclerotia in the field. And this is disease incidence. And so you can see that under 100 mycosclerotia per gram soil, there's very little disease. But once it crosses that threshold, there's a disease outbreak. And so during the time of inoculum buildup, growers and PCAs may miss the disease. And so preventing this inoculum buildup is key to managing your pathogen. Pathogens do not cause disease with a single attempt. Usually it takes multiple attempts. So you have to have a fair amount of the inoculum before you have successful infections. And infections may not always lead to disease. A plant can respond and suppress it. So it takes numerous amounts of inoculum and attempts and infections before you actually get disease. And in the case of verticillium, here's just a small table showing the differences in inoculum density and disease incidence. So for disease to be caused in strawberry, you only need 3 to 5 colony forming units per gram soil of Mycosclerotia, whereas I was just showing in lettuce, it takes more than 100. And so if you have low levels of the pathogen and you're growing lettuce, you may not know that the pathogen is there, whereas you plant strawberries and now you can see the pathogen because it's much more sensitive. And so this can obscure and confuse some disease diagnostic and processes. So once you have infected plants, you build up that inoculum with residue. And this becomes the main way that inoculum increases. You have an infected plant, it doesn't get rogued, it dies, the propagules reproduce on them, and now you have increased the amount of the pathogen in the soil. Residue also serves to protect the structures, the pathogen structures, from decomposition and from exposure to microbes and other organisms that accelerate the breakdown of them. And so they can live safely and protected in residue that's not decomposed, and therefore they survive longer in the soil. One way to reduce the inoculum buildup is to rotate with host-free crops. So these are non-susceptible crops. And this is typically part of a rotation. They can be economically viable crops, or they can be soil health, soil building crops like cover crops. But I do want to point out the importance of also recognizing that cover crops can be a source of host crops for a pathogen. So if we take verticillium again, this was some work that I did in my studies, identifying that there are numerous legume cover crops that support verticillium wilt, the pathogen, verticillium dahliae. And so, for example, if you have verticillium dahliae present in your field, these 4 cover crops, summer and winter cover crops, are not good choices because verticillium dahliae thrives in these plant tissues. And what's more challenging is that they don't show symptoms of the disease. And so they look like healthy crops, but in fact, the inoculum is building up in these plants and in your soil once they're introduced. So always be looking out for new updates and refresh your memory on what the host range is of these different crops. Our host range list is always expanding and evolving. So you have all this inoculum. It's not always realistic to rogue your plants. And so what can you do about that? Well, I'd like to introduce this concept. We have 2 fields. This is a known concept that I'll describe, in which you have 2 fields neighboring. You have the pathogen in both of these fields. In this case, it's Fusarium oxysporum, Formispecialis melonis, and you're growing melons in both of these fields. However, in this field, you get the disease, Fusarium wilt, but in this other field, the disease fails to develop. What's going on? In this specific case, this is a very famous area called Chateau Renard in France, where they have naturally occurring disease suppressive soil. And so the pathogen is there, the host is there, the environment is perfect, and yet disease fails to develop. And these soils exist around the world. They exist both naturally, and we've discovered that we can generate disease suppressive soils. And with modern technology, we're able to better understand some of the mechanisms behind disease suppressive soils. And there are several main mechanisms, and they are all microbial. So this first one is the generation of microbes that produce antibiotics. So you have the root surface, you have a beneficial microbe, in this case, Bacillus pseudolis, that's living at the root surface, the rhizosphere. It produces an antibiotic that then kills the pathogen. That's example one. Second example in how disease suppressive soils operate is by competing for nutrients. So soil-borne fungal pathogens are not motile. They survive as structures in the soil. And so how do they find plant roots? They find plant roots by exudates created by either the seeds or the roots that are planted. Those exudates stimulate germination and direct the pathogen growth to that seed or root. And so in this case, that's being demonstrated. This is for Pythium, which is an oomycete. But linoleic acid is the exudate that triggers germination. So if you instead have other organisms, other microbes competing for that exudate, these are all food sources, it interferes with that compound reaching your pathogen, and therefore your pathogen stays dormant in the soil and does not germinate and grow towards your roots. So competition for nutrients is a second way that disease suppressive soil occurs. The third way is direct parasitism. Many of you have probably heard of trichoderma. Trichoderma directly parasitizes plant pathogens. And the third way is induced systemic resistance. This is one of the plant defense mechanisms. And you have microorganisms living at the root surface that can stimulate a signaling cascade. And these signals can move to other parts of the plants that then create plant defenses, such as increasing the cortex of the roots to prevent successful entries. And it can also lead to the change in exudates from plant roots and lots of other mechanisms of defending plants against pathogen attacks. And it also leads to other signaling above ground. So those are four ways in which disease suppressive soils operate. And these are all microbially driven, as you can see. And so one way that we can promote microbial activity is by adding compost. So in this example, this was some research in which we added 4 different types of compost to 5 different fields. And CC is for Central Coast. These all took place on the Central Coast of California. And CV is the Central Valley, 2 different fields. And CCF is fumigated. So all the arrows are pointing towards fumigated fields. So they're fumigated with methyl bromide. And so what we did was we added compost and measured the microbial activity 2 weeks post incorporation. And you can see how the levels without compost were the lowest. The levels where manure compost and vermicompost were added increased them to intermediate levels. And the spent mushroom compost and yard trimmings compost had the greatest impact in increasing the microbial activity. We went back to those fields. They were producing strawberries 10 months later. So they had been undisturbed because they had strawberries planted in them. And this was the end of the season, 10 months later. And we could still have measurable increases in microbial activity. So this is a consolidated graph where you can see the fields with no compost added were the lowest. The vermicompost and manure compost were still intermediate, and the yard trimming and spent mushroom compost had the most, the greatest increase in microbial activity. This was just incredible to me that this small amount of compost we had added was able to continue to have measurable increases in microbial activity 10 months later. So we did similar work, but we did this in controlled pots. We added them with 20% compost, potting soil, but then we also introduced the pathogen, Verticillium dahliae, and planted strawberries into those pots. And when we removed those roots, we were able to see that vermicompost and manure compost had reduced the number of infections by Verticillium dahliae. And that's what this is showing here. In blue, we have the unamended control, so that was growing in potting soil by itself, compared to the addition of compost. Another great way to increase microbial activity is by plants. And this is part of the benefits of growing cover crops or any rotation crop, but keeping plants, the soil covered with plants, is all of the microbial food that's coming through these roots. So one way people describe plants and roots, or roots specifically, are as carbon pumps. They're releasing a significant amount, as much as 40% of the carbohydrates that come into that plants create are released through these roots. And that's all microbial food. That's sugar for the microbes. And so by growing plant roots of really any kind, you can stimulate microbial activity. So I'd like to pose a question to you to think about. With all of this in mind, let's take two identical fields. This is a hypothetical situation, just to stimulate some thoughts. We have two identical fields. They both have a pathogen at the same density. But you have one field in which all these soil health practices are being done. They're adding compost and growing cover crops and doing practices to minimize microbial disturbance. And the other field is not. So would you expect the same disease outcome in both fields? And so thinking about that and bringing that into a diagnostic situation, in a field with a disease outbreak, if you ask yourself, is it the healthy soil that has a disease outbreak, or is it an unhealthy soil that has a disease outbreak? Is it a disease outbreak, or is it a soil health crisis? And does this change your recommendation and management strategy? And if so, what tools might you have to evaluate soil health? So we did a study working with some organic farmers in our area, asking them how they evaluate for soil health. And they came up with three main metrics. The first one was plants. So looking at plant growth, they're looking at their main crop, but potentially more importantly, they're looking at their weeds, and they're looking at their cover crops and the plants that aren't managed with such care as their main crops as indicators of soil health. So they're looking at the crops that they're growing and the crops that they're unintentionally growing. Where they're looking at the presence and absence of biology. They're looking for biology. And so picking up a clod of soil and looking at it and looking for biopores. Do you see biopores? Do you see large biopores and small biopores? Is the soil holding together? When you dig a hole, how many soil critters are you looking at? And how many different kinds are you looking at? What are the indicators that microbial activity is there? Most of you are probably familiar with the soil food web, and we have this whole web of starting with microbial organisms that then feed on each other and feed nematodes, and it all goes up the trophic system to higher level organisms. And so as humans, we can only see these larger organisms with our naked eye, but the abundance of them and the diversity of the ones that we do see are indicators of the diversity and abundance of the microorganisms that we cannot see. So you can use that as a tool to tell you about the microbial health of the soil. The third one is looking at soil structure. And growers were looking at soil structure to indicate the soil health. They're looking at how the soil behaves when a tractor is running through it, by touching the soil, by digging small holes and looking at the profile, and looking at how water drains. And when it's windy, does the soil move? And so when we take these types of metrics that growers are using and apply it to our soil health triangle, I see some parallels. So we have our host, which is a plant, and the growers are using plants as indicators. We have our pathogen, but we also have the healthy soil biology that we're looking at. And then we have the environment, which is growers are describing as soil structure. And so it was striking to me to be thinking about we have our plant disease triangle, but the growers might be more thinking about it as a plant health triangle or a soil health triangle. So how are we creating these conditions for soil health? So in summary, I encourage you to think about how you can keep soil inoculum levels low. Roguing out plants, rotating with non-hosts, having good sanitation, and ensuring complete residue decomposition. And then thinking further about, okay, you have inoculum in the soil. How can you reduce the efficacy of inoculum, especially via soil health practices? So using compost and cover crops to increase the microbial activity. And that in turn will encourage complete residue decomposition. It will accelerate the inoculum attrition rate. It will increase microbial competition, increase antimicrobial compounds, promote plant defense mechanisms, and overall increase plant health. So thank you for your time, and I will take questions or pass the mic.
Thanks, Margaret. Let's launch the poll questions. And if you're going to request the CE credit, make sure that you answer these poll questions. Here we go. And I did on accident say DRP instead of DPR. I apologize for that, but the credit is through DPR, Department of Pesticide Regulation. Okay. Why don't we close the poll and share the results? And Margaret, if you could go over those for us.
Sure. So what stimulates the germination of most pathogen Mycosclerotia? Root exudates, excellent. And beneficial microbes can suppress soil-borne pathogens through multiple methods. Which one is not true? So they can induce plant defense mechanisms. They can parasitize plant pathogen like trichoderma. They generally do not cause uncontrolled cell growth, so that is not correct, or that is the correct one. And they can produce antibiotic compounds. So the third one. Great job, folks.
There's a couple requests to maybe throw up that last slide that has the list of actions to take and the 4 ways to suppress disease or to make disease suppressive soils. Do you want a quick
This one?
Yeah, I think so. Is there a way to summarize those quickly?
Sure. So several of these here. So the bottom 4 are specifically thinking about the disease suppressive soil. So how microbes can directly antagonize soil-borne pathogens through microbial competition, by creating antimicrobial compounds, by promoting the plant defense mechanisms, and by increasing plant health. And I added the first 2 because microbes are responsible for decomposing plant residue, and that's critical to exposing those soil-borne pathogens that can be tucked away in the plant residue, exposing them to the microbes that can then act on them to break them down, which is also related to number 2, accelerating inoculum attrition. You often hear that some soil-borne pathogens can live in the soil, survive in the soil for 5 or 10 years, and that's a long time to have to rotate. So how can you accelerate their breakdown? And the best way to accelerate that is by microbial decomposition. So having microbes that can actively work on them and break them down.
Perfect. Thanks so much. If you want to put your question in the chat, I promise we'll get to it, but I'm going to turn it over to Martine and have him share his.
Again, hello, my name is Martin Garena, and I'm with the National Center for Appropriate Technology. We manage the ATRA project, which is a sustainable ag information service, full of wonderful information, publications, videos, blogs, webinars like these, etc. So check us out online. Anyhow, so I'll talk today about solarization and biosolarization, harnessing the sun to control pests. So first off, let's talk about solarization um, and what it is. Basically, it's passive solar heating of the soil induced by covering moist soil, or I'd say wet soil, with transparent plastic. Okay, there's a lot of misconception thinking that black plastic works as far as heating up the soil. Well, it does a little bit, but nothing like the clear plastic does. It's like a greenhouse lets in the ultraviolet rays, and then you have the moist soil, which carries the heat or transfers the heat a lot easier than, let's say, dry soil. Then it traps the outgoing radiation and keeps it warm for longer periods. So you have days of the sun hitting this plastic little mini greenhouse over the soil, and it heats it up pretty well. So what this does, temperature-wise, of course, it inactivates the soil pests and in some cases remediates toxins and releases nutrients. And we'll see a little bit more how this takes place. So this is kind of a graphic from UC Extension on gardening, but it can work on farming too. Basically, you till the soil, you smooth it out, even it out. The two things that are important here are getting the sufficient amount of moisture, as much as you can actually, and being able to go back in and put the plastic down and sealing the plastic all around the edges so that temperature and gases do not dissipate or escape. So there's two ways of doing it on a large scale. You could easily do it by beds with a roll of plastic and a plastic mulch setter or spreader, or flat fume, which just is this is 1 acre plot that you put patches of plastic together and then put them roll up the ends and use these like giant metal clothespins, if you will, to seal everything up and get a uniform um, fumigation, if you will. I like this method better, the flatbed. This one, of course, you have to do every year because you have the weed seeds and propagules and everything else in the row that will come back next year and reinfest the area that was already treated. This was an experiment that I did with the city of Davis. This was a restoration of native grasses. This was about 12 years ago. Um, to this day, it's still predominantly native grasses. Of course, you get some weeds that fly in or weed seeds, but then they've been taken care of. So it's like a nice acre stand of native grasses and wildflowers. This was an experiment done on campus at UCD by Jesus Fernandez Bayo, where he solarized these beds and he had these, fancy instruments with different monitors that kind of monitored the temperature um, at 5 centimeters or 2 inches and close to 13 centimeters or 18 inches. And you can see the fluctuation of temperature. The bottom axis is days. So, it would drop down. Equivalent temperatures in centigrade 50 is 122, 40 is 104, and 86 is 30. So you can see the fluctuation day and night at 2 inches and also at 5 inches. So even at, 8 inches, you're still getting, high temperatures of at least, 90 degrees, 95 or 85 degrees or so at 8 inches. So I did a similar experiment with that flat fume, but used a really cheap cooking thermometer And you can see the flat surface temperature. This is on the surface of, 100 and close to 130 degrees. Ambient temperature being 93 degrees. This was a thicker plastic. It was like 4 mil, but we were able to reuse it in other places after. So at 3 inches, same time, we had 111 degrees Fahrenheit, and at 6 inches, close to, well, 100 degrees. So it gives you an idea. And this was at about 11 in the morning. So the heat of the day at 4, 5 in the afternoon in the summer, and this was in June, you're getting higher temperatures. So we did this for a month and got great results. Limitation of this here in the Central Valley, it takes about 4 to 8 weeks, depending on where you are and your soil type and how wet your soil is. Also, you have certain pests that are heat tolerated. In the case of weeds, you had um, uh, field bindweed and things like Bermuda, which on the surface will die, but then you have the rhizomes that are buried deeper that can't survive and come back. Of course, it's all dependent on local climate and weather, and depending on your soil type and how well you got the soil wet is limited control depth. Another disadvantage also of biosolarization, well, solarization is the plastic. Plastic right now is a big problem, pollution wise. What we did that flat fume, we recycled it and used it in other places at other times. And some farmers have done that with greenhouse plastic that I've worked with. So biosolarization is the same process as solarization, but you add organic matter. By adding this organic matter, the microbes, they'll go anaerobic and will perform what's called anaerobic soil disinfestation, or ASD. And ASD basically is a process of disinfesting the soil by creating anaerobic conditions with the incorporation of organic matter on, very wet soils, then covering it with a sheet of impermeable plastic to prevent oxygen from diffusing into the soil. These are experiments done on the coast where it doesn't get very hot in the summer. It's relatively cool. So of course they use black plastic. There's no sense in using the clear plastic. I've tried using black plastic and I mean clear plastic in Salinas, and it doesn't get hot enough, and you get all the weeds growing up into the plastic like a greenhouse and actually kind of lifting the plastic up. So in this experiment was done on strawberries. They basically just got it anaerobic wise and causing the microbes, primarily bacteria, to break down the organic matter and produce biotoxins or biopesticidal products. And this takes anywhere from 2 to 5 weeks to accomplish. Then biosolarization basically is anaerobic soil disinfestation, but with transparent plastic. So mixing solarization with anaerobic soil disinfestation. This accelerates the process 5 to 10 days. So the mechanisms that inactivate the pest can be the temperature increase, where you saw in solarization, they can get pretty high, especially on the surface. Oxygen depletion, especially if you get the soil saturated, you have enough water there. And that causes a lot of these microorganisms that are known as facultative microorganisms because they can consume oxygen. And then once the oxygen is cut off, they start consuming carbon and eating up the organic matter, and releasing numerous biotoxins, such as fatty acids, acetic acid, which is vinegar, propionic acid, butyric acid, valeric acid, capriotic acid, as well as aldehydes, alcohols, ammonia, metal ions, and volatile organic products. This, of course, depends on your biomass source. What are you using? In some of the trials I did, I only had access to compost, but a lot of Researchers are looking at getting rid of organic waste on a farm, for example, almond hulls or walnut hulls, pumice from processing facilities, etc. As long as it's organic. Also, uncomposted green waste works really well. So this is just a graphic of on a scientific journal of the process. You see the ultraviolet rays going in, heating up, the temperatures go up, pH drops, of course, becomes acidified. The organisms start producing these biotoxins, and everything is trapped within the plastic that's sealed well and moist enough to become anaerobic. So the advantage of biosolarization is a shorter application time. It could be anywhere from 6 to 10 days, depending on how hot it gets and where you're at. You recycle agricultural wastes. Um, it's more effective in cooler or less sunny areas. It improves soil quality because of the added organic matter. And it's effective in deeper soil layers and can begin immediately. For example, if you use a cover crop as your organic matter source, you can mow it down or incorporate it in the soil and just go right back in and well, irrigate it, make sure it's wet, and tarp it and cover it up. This is a experiment done, I think, in Spain on strawberries against nematodes, where they had, you see biosolarization there in the middle against root knot nematode and then against the soil lesion nematode. Keep in mind the letters next to the product indicates the similar letters indicate there's no significant difference. So you see biosolarization is just as good as the top other three chemical disinfectants against the root knot. And same thing with root lesion nematode. Whoop. And this is another trial that was done in Spain with verticillium and rhizoctonia. And basically, they're using fresh sheep manure. That's another thing that could be done here, but there's a big taboo against using, due to food safety concerns of using manures. But there's no problem with it. I mean, you reach temperatures high enough to get rid of all the possible pathogens such as E. coli and Listeria and so forth. So looking at this, you can see that they used fresh sheep manure as well as beer bagasse, broccoli residues, and combinations. And again, the letter, the control on the bottom indicates that was infectivity or the death of the plants, of artichoke plants in this case. So control with the verticillium killed, 100%, and with the rhizoctonia 55 to 77 or 78%. And then if you go up the list, you see that all the treatments were significant. The fresh sheep manure actually seemed to have done better all by itself. And then there's two depths there, the 15 centimeters and 30 centimeters for both. I want to talk a little bit about soil steaming, which is also heat related. This was this machine I saw in Salinas that injects steam. It's like a locomotive, actually. It's huge. It's very expensive. It's over $500,000. That it goes through each of those rows and injects. There's these shanks in there and injects steam at about 3 to 4 inches into the soil. Keep in mind that steam, water boils at 212 degrees Fahrenheit. So the steam is probably hotter than that. It goes through each row, injecting the soil. And you can see it kind of, people are touching it. It's really hot at the touch and it's still releasing steam easily, after at least an hour. And it's still warm, but it gets really hot initially and it's somewhat effective. On a smaller scale, there's this individual row steamer. This one's worth, I don't know, about $40,000 or so. Um, it relies on this hose that releases the steam throughout the area that it's laid down on, and then you cover it with this impermeable black top. And here's a shot of a small farm in Sacramento where it steams um, one row at a time. It takes about I'd say about 20 minutes, and then the crew has to move the line and everything once it cools to the next section, and you can see how well it's killed off the weeds. And the farmer in this case had a particular problem with sclerotinia, or lettuce leaf drop, which was a big problem in his production system. He grows a lot of lettuce under greenhouses. And you can see before he would do this, he'd spend a lot of money weeding as well as losing easily 20-30% of the crop due to the sclerotinia. And now it's like no weeds at all and almost 100% harvest. So, what about the beneficial microbes? People always say, oh my God, you're going to kill them off. Well, a lot of beneficial soil organisms are able to survive solarization and recolonize the soil quickly afterwards. Keep in mind that they're mobile, and a lot of times they're down in I call it the Goldilocks zone, where it's just right, it didn't get too hot, and they go back, and of course, a lot of their friends and family died and are there, organic matter, and they consume them, and it causes a lot of a release of nutrients, and actually a lot of times the plants grow quicker, and there's a lot of nutrients available. And some microorganisms, such as those found in compost, can tolerate heat, and they're the first to come back and recolonize. Things like mycorrhizal fungi also. And I've had the experience of the tilth in the soil is a lot easier to work after the solarization process. So in conclusion, biosolarization is as effective as solarization in less time, 1 week instead of a month. Now this is in the valley. Of course, I can vary where you're at. And then for biosolarization and solarization to be effective, soils must have enough moisture, and the edges of the plastic have to be sealed to prevent moisture, heat, and gases from dissipating, plus anchoring the plastic. And then, while this, I was talking about weeds in particular, those weeds that are not controlled or diseases in this case, have to be dispatched by other methods. In the case of weeds, such as mechanical controls, part of IPM program, as well with diseases. Let's say you have to probably use resistant varieties or something that's tolerable. So
Thanks so
that's it.
much. That's great.
Yes.
Let's launch that second round of poll questions. And if you have any questions, feel free to put them in the chat and we will get to those next.
So yeah, looking initially, soil temperatures are definitely the main reason. In the case of solarization, okay. Um, and the goal of solarization, biosolarization is pathogen, yes, and weed reduction, yes, as well as nematodes. Well, I guess they're considered pathogens, and any insect that might be there. A lot of insects pupate, and of course they'll probably fall victim to the heat. Um,
Excellent. Great.
yeah.
Thanks
So
for
are
those.
there any other questions?
Yeah, there's a question about, do different kinds of cover crops yield different results, weed suppression, pathogen suppression, when used in the biosolarization process?
Mm. Well, yeah, organic matter sources. In the case of cover crops, there is research being done with specific varieties of cover crops. Like I mentioned, each source of organic matter will release different type of bio pesticides, if you will, of gases, so forth, depending on what they are. But right now, I think they are focusing in on certain types of cover crops and what kind of control are they're getting from with certain types.
BJ Mills has their hand raised.
Hello. Yeah, I was wondering about when you mentioned the solarization, can some of the benefits of releasing nutrients. I'm just wondering if you can expand on, I think you kind of alluded to there, perhaps some of those dead microbes. Is that where
That's
that's
it. Yes, that's exactly it.
okay?
Um, the same effect occurs with chemical fumigation, like when methyl bromide, it'd kill off mostly everything. Um, and you'd get this boost in growth and development of the plants. And of course, it is the microbes that were there that are deceased, and then the other organisms that come back recolonize and release a lot of these nutrients. Um, um, and like Margaret mentioned too, a lot of these organisms are mobile, whereas the pathogen resting structures, sclerotia and so forth, are stationary. So they're basically, you know, um, it's difficult for them to repopulate the area.
I have a question. I was wondering, Margaret, a couple of things. If you could tell us where we could find a list of cover crops that foster pathogens. And then also, can you discuss how just because a cover crop is on that list doesn't mean you shouldn't ever grow it. You should only, because they're good plants in certain situations, right? Thank you.
Yes, thank you for your question. So I'm not aware of like a single consolidated list. I know that Margaret Smithernople from the USDA in Lockford has a great list and does have comments about their relationship to diseases. Um, but it's worth, and to your second point, doing like a individual and specific kind of check. So for example, your point is correct. These are very specific relationships between which host plants host what pathogens. And so first of all, it really encourages people to get a correct diagnosis and not a guess about what your disease is because it has huge impacts on your rotation choices. So get your plant sample sent to a diagnostic lab and confirmed. It's very difficult, even for experts, to use visual diagnoses alone to differentiate between Fusarium and Verticillium wilt, for example. So get a diagnosis and then use that to kind of cross-reference the plants that you're looking at growing. And I would only caution you against growing host plants if you have the known pathogen. Otherwise, these plants are wonderful to grow and have a whole host of benefits. So it's very specific to the problem.
There's one other question, Margaret, for you about the omeocytes and if they are Phytophthora and Pythium motile, are they motile? And then can they move away from suppressive soils or towards hospitable environments?
Yeah, so those are both oomycetes, and they do have flagella, and they are, when under wet conditions, they can swim, so to speak. And that's why they're often associated with wet environments. Well, suppressive soils, this tends to be happening at a greater scale than in little like pockets. So moving away from it is not really realistic because we're talking about something happening at the field scale. So no, I don't think that that's plausible.
Right.
And I also want to highlight, sorry, that if you remember those four examples of creating antibiotics and interfering with exudates and plant defense hormones, those are often all happening at the root interface. And so it's happening where the plants are. And so it's kind of like an in situ extended defense mechanism of having microbes that are living and thriving in the rhizosphere that are protecting the plant from outside attacks of the roots. So it's closely associated to wherever plants are. And yeah, so I hope that helps kind of clarify and think about this system.
I had a question for Martine. Does the BIOS or the ASD biosolarization, does that work on really light soils, sandy soils? Because the soil, you have to saturate it and it has to become anaerobic. So will that occur in sandy soils?
How much organic matter is in it, and how much water it can retain. Obviously, if it's just like sterile sand, it'll just filter right out. You'll get a lot of airspace, and therefore, less transmission of the heat, and yeah, less organic matter activity. I've seen some, what would you call it, silty soil, I guess, between sand and clay, where it works very well. But sand itself, I think it'd probably be more difficult, if not patchy, if at best.
Thank you. One more question for Margaret. The induced resistance is so fascinating. Is there much known about that, or how can we learn more about that?
Yeah, there's a lot known about it. Induced systemic resistance is a general term you can search. Plant defense mechanisms. I have a great two-part series on my website or on my YouTube channel. You can find it, Margaret Lloyd, UC Cooperative Extension YouTube channel. It's a two-part series all about plant defense mechanisms. And it's part of the organic seminar series. So you can look for it there. I think that's probably a really nice summary. And there are companies that are trying to create compounds to induce plant defense mechanisms. So that's a product on the market. This is another way that people might hear about it. It's trying to tap into the plant defense mechanisms and give operators more control over stimulating it. But at this point, the best way and our best understood way is to stimulate the microbes in the soil, and they will do the work for you.
Excellent. Thanks so much for sharing those resources too. We can definitely get those up on the link for the resources for this webinar as well. I want to thank Martina and Margaret for sharing their knowledge, and thanks to everyone for attending. We'd love to have your feedback, and if you need those CE credits, we need your license numbers, and the DPR credit requires you take a quick 10-question quiz. There are additional resources on the website that are recommended by Margaret and Martine to check out, including a case study of 13 organic farms in Northern California on their understanding of soil and soil management, plus the ATRA publications on soil solarization and biosolarization. And we hope to see you at our next webinar on December 10th next week. Thanks so much for attending.